Somehow the sequence of bases in DNA has to be converted into a sequence of amino acids in the protein. Now, the function of a protein is dependent very much on its shape, and the shape of the protein is dependent on the sequence of the amino acids and the primary structure. So we've got a problem there, though, because, of course, we have only got 4 bases in DNA. We've got A, T, G, C. But, of course, amino acids, there are 20 different amino acids. So that means that it can't be 1 base per amino acid. It can't even be 2. It won't be enough. The minimum number you could get away with is 3 bases coding for 1 amino acid, and that is actually what happens. So, for example, if you have the base sequence in the DNA of T, 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 that will code for the amino acid phenylalanine. Whereas if you had, say, GCA, that would code for the amino acid alanine. Okay, so there are 2 different processes in this. What's going to happen then? First of all, the DNA contains a sequence of bases. Well, in eukaryotes, that's in the nucleus, and prokaryotes, of course, it's all in the same compartment. But that needs to be, you can't read directly from the DNA. What happens to happen is a copy of the DNA, the base sequence in the DNA, has to be copied into a molecule of RNA called messenger RNA. In eukaryotes, that messenger RNA then passes out into the nucleus, and it is converted at the ribosome into a sequence of amino acids in the protein. And this is called translation, this process, and we'll talk about that in the next video. It's a bit much to do in one. This process here of making a copy of the DNA is called transcription, and that's what we're going to talk about now. Okay, so transcription then, to make the messenger RNA molecule. Okay, so we have our double-stranded DNA here, and don't forget the strands run anti-parallel, so that's the 3' running 5'. This other strand will be 5' to 3'. The numbers 3 and 5 refer to the N-carbon atom in the sugar. That's what it refers to. And what's holding those 2 strands together, of course, is our hydrogen bonds between the base pairs G to C, T to A, of course. Okay. Now, before transcription can occur, you have to break these hydrogen bonds. We've seen the 2 strands. So if I draw that, we get a little bubble forming in the DNA there. Okay, well, the hydrogen bonds are still intact here, but in this part, they've all been broken. Okay. Now, we need to make a messenger RNA, complementary messenger RNA to this strand here. Okay. So, but before that can happen, well, we need the monomers. And the monomers that are used are the nucleotide triphosphates called NTPs. Okay, so for example, you're going to have 3 phosphate groups, and then you're going to have a sugar, and then the base, sugar, and then you're going to have the base, whether that is AT, right? So you can have ATP, triphosphate, guanosine triphosphate, cytosine triphosphate. It's not thymidine, it's uracil in RNA. You don't have T in RNA, so it's going to be UTP. Right. Okay, so what's going to happen? Let's say we have a base sequence there for sake of argument. Let's have like G, C, T, A there. Well, we've got this little nucleotide triphosphates. Okay, so we have that. These are all separate. That's one with an A in it. G, and there's lots of those about. That's one with a C in it. So they're going to hydrogen bond to the complementary base. So there we are going to get a C forming. Across this one, you've got a G. We'll have an A there, and we won't have a T there, we're going to have a U. Right, they're not joined together just yet. Okay, so then the next, what actually happens then, how that works is, if I just rub that there, imagine you've got the end, this is the end nucleotide in the RNA, so you're going to have, that's going to be phosphate, sugar, and the base. Right, and you've got a free OH group here on carbon atom number 3, and what happens is that phosphate sticks on there, so, and then these two phosphates get cleaved off, and you've added another base onto the chain, so it's one longer now, and then the process carries on. Okay, now what, of course, that's not going to occur without an enzyme. I'll join these up because they've all been joined up with an enzyme. The enzyme required for that is called RNA polymerase. There's a yellow blob. So we keep sticking a new nucleotide on the end of the RNA, and that will work in a 5' to 3' direction. So that means this strand here must be the opposite way around. Of course, that's going to be 3' to 5'. So we draw what happens a little bit further down then. So we've now got our bubble in the DNA, double helix, separated, and we've got, this is RNA which had been made before, and this is RNA currently being made, and we've got RNA polymerase on the strand there doing its job. Okay, what's actually got us, before that can start, before the RNA can be made, you need a start codon there. So this basic, when you have 3 nucleotides, okay, so for example, I said before phenylalanine was TTT. That's called a codon. Well, in the DNA, it's not strictly called a codon. It's codon when it's in the RNA. But then you've got this. They're coding for phenylalanine. What you will have is something called a start. So one triplet, and it's actually ATG on the DNA. This is the start. Okay. So when RNA polymerase sees that, it will start making an RNA molecule. It's actually not quite sufficient. You need a little bit more than just this sequence in the DNA in order for the RNA polymerase to work. You need some special sequences upstream of that, people usually say. You need some special sequences there to help the RNA polymerase bind. Then it will start making the RNA. Note that when you're making the new RNA strand, you're using nucleotide triphosphate, so ATP. And remember, ATP is a high energy molecule. You need respiration to make ATP. It's quite, so this is, and it's the same for all 4 of the nucleotides. They're all quite high, they've got a lot of chemical energy stored in those molecules, and so you need a big investment in terms of respiration and so forth in the cell in order before RNA can be made. Right, so the RNA molecule, once it's eventually it will drop off the DNA. It's finished, and we've got this is our RNA. We'll call it the first transcript because it's not quite ready to be a messenger RNA just yet. Right, now in prokaryotes it is ready, and it's ready to be translated and to make a protein chain from it. And of course, in prokaryotes, it doesn't need to pass out of the nucleus because there isn't a nucleus. Everything occurs in the same compartment. But in eukaryotes, this first transcript isn't ready. In eukaryotes, I'll draw it. You have these red bits are coding regions. They actually code for proteins, and these blue bits don't code for anything. They're non-coding, so they're bits of the RNA copied from the DNA, and they're called exon sequences. Whereas these ones which do code for things are called the introns. and those exons have to be spliced out. Okay, so what happens is that goes from there to this, and the exons will be cut out, and so the blue bits will be thrown away. You end up with the pure just coding mRNA. So this is the mRNA, which can then, in the eukaryote, can pass out of the nucleus and bind to the ribosome where protein synthesis is going to take place. And we'll cover that in the next video.